All right, so here's our lecture. We're going to combine oncolytic viruses and gene therapy. And this is a lecture that I really wish, I guess now that we did this early in the semester, because this is one of my favorite topics. I used to work in a gene therapy lab. I worked with adeno-associated viral vectors. They're called adeno-associated virus because these viruses were found as a contaminant in an adenoviral prep. And so I worked for a number of years making gene therapy vectors. So using viruses for therapy is a cool thing to do. You can take something that's normally destructive and you can pull out the pathogenic sequences and put in something that's therapeutic. Or you can modify something that's pathogenic and make it work in an oncolytic. Or there's a lot of different approaches you can take. Now, there's kind of a reason we're looking at things in this order. So we're going to be talking about influenza coming up, and we just talked about pox viruses, and using viruses to treat cancers has an interesting history. So scientists who were treating people with cancer began to notice over the years that if someone came down with a case of, say, influenza, that it might put their leukemia into remission. And so the idea that a viral infection like influenza could actually be beneficial in treating cancer, that idea has been around for a long time, but it's not until very recently that we've really had the technology and the science to do it in a safe and effective way. Some of the early methods that people did weren't they would not go by review boards today. So today we've got a lot of regulations and a lot of hoops that you have to jump through before you put a virus into a person with good reason. You want it to be safe, and so some of the early experiments they did you wouldn't be able to do now. So if we look a little bit at the history here, excuse me. So, they've been using chemicals for treating cancer for a long time. So the nitrogen mustards, essentially using mustard gas, using toxic chemicals in cancer therapy for a long time, using radiation, which we don't use mustard gas anymore, but we still use radiation, though we fine-tuned it. It used to be, like if you had cervical cancer, that they would just shove a piece of radioactive metal up the vagina, up near the cervix, to try to kill the cancer cells, which, obviously is not such a great idea. Now, they do it in a much more focused way. And the first oncolytic virus that was approved for use was in 2005. So virotherapy is really using any virus for a therapeutic benefit. So we're going to start off talking about oncolytic viruses, but you could theoretically use viruses for other benefits, like phage therapy. We've talked about before, using a bacteriophage to target bacterial infection would be a form of virotherapy. So using oncolytic viruses to destroy the cancer cells without harming the healthy cells, this is our, this is the $1 million question. This is our the big goal. So there's a couple ways that we can target cancer cells with viruses. So viruses need really active, metabolically active cells to replicate, and tumor cells are much more active than our normal cells. So targeting cells and being able to replicate in cancer cells faster than they could in healthy cell. Inducing apoptosis. Now with this, obviously, you really want to make sure you're only infecting the tumor and you're not infecting the healthy cells because you're triggering cell death. Introducing of, this is lysing the cancer cell, and yeah, the way that they phrase this in your book is a little odd. So allowing the cancer cell to die after the virus is replicated, then the virus can spread to other tumor cells. We'll get into this a little bit more. But basically, all these three things come to directly killing the cancer. And then this one is slightly more complicated. Here you are having the virus replicate in the cancer cells, and then that attracts the host's immune system. So the cancer cell is going to, display the virus on its surface, on MHC1, for example, and then a cytotoxic T cell can come along and recognize that and then kill the tumor cell, for example. Okay, so we've got a couple different illustrations here, giving you the overview. So how do you make sure that the virus is only going to replicate in cancer cells? This is pretty tricky. So there's a few different mechanisms you can use, and sometimes you might use more than one of these mechanisms. All right, the first is you look at the transcriptomics, or which transcripts, which mRNAs are being produced that are different in the cancer cell versus a normal replicating cell. A normal healthy cell. So the healthy cells here are on the bottom, and the cancer cells here are on the top. Okay, so targeting transcription. There are some genes that are turned on only in cancer cells, right? And some viruses are dependent upon those genes. So if you use a virus and you inject it, say you give this intravenously, and the virus might get into lots of different cells, but the virus can only replicate in cancer cells where the genes it needs are turned on. All right, so that would be targeting transcription. All right, the second way we have here, all right, is by targeting attachment. So some cancer cells express unique antigens, or express unique sugars, surface proteins, or perhaps they upregulate them to such a high degree that you are going to find 90% of all of that surface antigen you're going to find on a tumor cell or a cancer cell. So again, if you injected this intravenously, the virus is going to go all over the body, but it's only going to bind and attach to the surface molecule that is uniquely expressed on the cancer cell. So the virus can enter the cancer cell here, but not here because it doesn't have the right receptor. And this is what I specifically did a lot of work on, is looking at the attachment of our viral vectors to their specific host cell receptors. Now, then we've got the next one here is exploiting the innate immune system. So you've got the antiviral state, Remember, interferon is really important for triggering that antiviral state. Now, in a normal cell, you've got interferon signaling. Interferon signaling is going to stop the viral infection. However, in a cancer cell, cancer cells typically downregulate interferon. And so if there's a defect in the innate immune system, if there's a defect in interferon in the cancer cells, then the virus can only replicate in those cells without interferon. So the virus can only replicate in the cancer cells because it doesn't have a fully functioning antiviral state. It doesn't have good interferon. And the last mechanism is timing of cell death. So, and this one is a little counterintuitive. All right, this one sounds a little odd. But, okay, so you've got a viral infection with lots of different cells, right? And say these are normal, healthy cells, and then in orange here, we've got some tumor cells. All right, now we've got the virus. The virus will be in light blue. All right, so we've got our virus. And in normal cells, the virus infects, and then the cell can detect that it's been infected by a virus, and the cell is going to undergo apoptosis. So the cell is going to kill itself, and the virus doesn't get to completely replicate. And so all of these neighboring healthy purple cells don't get infected because this one cell committed apoptosis, committed self-suicide, and then the viral infection couldn't spread any further. However, many cancer cells have defects in the apoptosis pathway, and many cancer cells turn off apoptosis so they can replicate uncontrollably. So a virus gets into a cancer cell and apoptosis is not inhibited, so then this virus can spread and grow and replicate, and then it can spread to the other cells. And then when the virus spreads, then it winds up killing these tumor cells, and all these tumor cells get killed, but when they go to spread to healthy cells, the infection can't spread far into the healthy tissue. So these are 4 ways that you can target cancer cells and not get the healthy cells. Now, it's not uncommon to use more than one mechanism here. It's also not, it's also important that whenever you use virotherapy, that you can treat the virus with an antiviral medication. So if things don't go according to plan, or for whatever reason you need to stop it, you can give the person an antiviral medication, and that will stop the virus from replicating any further. That way you don't accidentally get the person sick. So this is kind of a condition now for using virotherapy, is you have to be able to stop the virotherapy if you need to. So what are some challenges in virotherapy? And, this isn't just virotherapy, but this is also gene therapy. All right, so one big problem here is immunity. If the immune system mounts a response against your virus, so you've got a virus here with very helpful genes, But if your immune system targets it, then you can't get your viral vector or oncolytic virus to where it needs to go because the immune system took it out. All right. Then you've got delivery. How are you going to deliver this? Are you going to deliver it IV? Are you going to have to inject it directly into the tumor or the damaged organ? Are you going to do this outside the body? Maybe you're going to do this ex vivo. How are you going to deliver it? This is really, really important, and each of these methods comes with a challenge. IV, for example, if you administer any viral agent IV, within an hour, 90% of it is going to go straight to the liver. And this is actually a good thing if you're treating hepatitis, but if you're treating anything else, then almost all of your therapeutic virus has gone to the liver and not to whatever other organ you're trying to target. That's the liver's job, and it does it very well. If you're injecting it directly into the site, this could be problematic depending on where you need to inject it. Some problems with IV and injection are the brain barrier. So there's a really good viral virus file in your book talking about the blood-brain barrier. And so if you have a brain tumor, you can't give the medication IV because the blood-brain barrier will block it. None of it will get into the brain where it needs to go. So that means you need to drill a hole through the person's skull and to inject it into the tumor, which is obviously not something that's done lightly. You can use biomarkers to track the progress of delivery or infection. Biomarkers might be something fluorescent, for example, but often if you're tracking the progress of the tumor, you're looking for the tumor to shrink. So sometimes to counteract the host immune response, you can try to combine viruses. So you can try to provide one virus that's going to help against the immune system and another virus that would directly kill the tumor. Though when you're combining viruses, this does up the ante because you have to prove that both of these can be safe. You have to look at the safety profile of both of these. And whenever you're dealing with the immune system, you really have to be careful because you don't want to overstimulate the immune response and possibly send the person into shock. And you want to make sure that... Okay, sorry, I am being summoned and we're almost at 20 minutes. So we will stop here for this is part 1, and then we're only going to have a part 2. So this lecture is just going to have 2 parts, and both of those will take some material from the gene therapy and from the viruses and cancer chapters. Right, so I mentioned briefly the very last thing I'll mention here, safety. Right, you don't want to overstimulate the immune system. You don't want to send a person into shock. It hasn't been 2 minutes, honey. It's been like 30 seconds. Give me another 90 seconds, okay? All right, so safety. You also want to make sure that that the person might not cross-react with something. So if a person is being administered a virus that contains a therapeutic gene, you don't want their immune system to mistake that for something else and perhaps overreact, right? You don't want to have an allergic reaction to the virus. And of course, you have to make sure that the viruses are only harming the tumor and they're not replicating in the healthy cells. And you also have to look at integration events. So we talked about in the last lecture how oncogenic viruses can integrate. And if they integrate into a region, into the promoter region upstream of a proto-oncogene, they can turn that oncogene on, and that leads to uncontrolled cell growth. So you really need to be aware, and you need to eliminate any chance of integration. So there's a lot of safety concerns. So when you have this possibility of using two viruses, you have to make sure that both of them are equally safe. All right, we'll talk more about some trials in the next lecture, in the part 2.